Let's now move on to the final category of catalysts, which is your biological catalysts, in other words, enzymes. So the bio students should be familiar with this. So enzymes are biological catalysts which have a very specific activity. So these are catalysts, so obviously they will speed up certain chemical reactions. Now it's important to note that without these biological catalysts, okay, which are usually in the form of proteins, we won't learn about proteins in our syllabus. So if you want to know more, you can go and learn from the bio students about what proteins are. The most we'll touch about is later on when we do organic nitrogen compounds, okay, about the formation of proteins from amino acids. That's about it. Okay, you want to know more, you go and learn from the bio students, and of course the biology students will know much about this already. Now, these proteins will fold into certain specific shapes, okay, in an activated form, which will enable them to act as enzymes. So as enzymes, they will then be able to catalyze reactions, because otherwise, without them, most biochemical processes will be too slow. All right, we won't be able to live. Life cannot be sustained. Furthermore, enzymes are also important in many applications. So these are just a few of them. It's not an exhaustive list. There's more actually. Okay, now, where do enzymes fall? Are they homogeneous or heterogeneous? Previously I said homogeneous. Actually, that's not true. The problem is with enzymes as biological catalysts, they occupy a middle ground. Okay, they can be homogeneous. Okay, let's be a bit more specific. Okay, homogeneous or heterogeneous. Right, not just because they're colloidal in nature. You want to say, what does colloidal mean? It means dissolved in a larger form. It's that they can be dissolved. Sometimes they can be dissolved. So dissolved in the stomach, for example, your stomach has enzymes which help to break down certain types of foods. But they can also be on the cell membrane. Okay, so when they are in the cell membrane, will you say the cell is dissolved in the water? No, you can't. You wouldn't say that. So it's in a way it's a solid form, right? It's a membrane. Okay, so in a solid form, then in that case, when it works on chemicals in solution, then of course it's heterogeneous. So they can be both homogeneous or heterogeneous depending on the situation. Now, these enzymes are very selective. They have very specific activity. They work only on a few chemicals, on a few reactions. Okay, so they only tend to catalyze a specific reaction or type of reaction. Very important. So they only work for that purpose. You try to make them work for something else that's not going to work. And of course, being proteins, they are very sensitive to temperature. Right? Most have optimal temperature of about 38 to 40 degrees Celsius. You may realize, hey, wait a minute, that's just about our body temperature, and that's correct. So at lower temperatures, they are not as effective, okay, because generally there's not enough energy for them to do what they need to do. And at high temperatures, they will just be destroyed. Okay, as to how and why they're destroyed, again, you can go and find out from biology. Go and read up the text. So here's a graph that shows how enzymes, how effective they are in terms when the temperature changes. Right, so most of them are effective at 37 degrees Celsius. Okay, you know our body temperature is what? 36.9, yeah, around there. Not only that, not only are they sensitive to temperature, they are also sensitive to pH changes and poisons. Okay, there are a lot of things They are sensitive to metal ions, okay, other chemicals. A lot of things can mess around with enzymes. Okay, so example, when we ferment wheat or barley to form alcohol, okay, barley lah, let's use barley as an example, to form beer, right? So ethanol will poison the ethanol that's formed as the enzymes ferment the sugar into ethanol. But the ethanol, the product itself, will also destroy the enzyme, especially when it reaches a certain concentration. Then the fermentation will stop. Okay, so this is just an example of the sensitivity of enzymes. Now, like all catalysts, they catalyze reactions by providing an alternative pathway with a lower activation energy. And how enzymes do this is they will, what we call, form a complex. They will latch onto the reactants. Okay, they'll latch onto the reactants. Then they'll do things to the reactants. Okay, so do things to the reactants already. The reactant has changed to a product, then they'll leave the product. Okay, then they'll detach itself. So this is the sequence that's shown. So it forms a complex. And over here, you have your bonds broken, bonds form. Then, after the product is formed, it detaches from the product. So this is just a very simple, very simplified explanation of how it works. Now, you may consider, okay, so how does it pertain to kinetics? There is a very specific implication of this mode of action. Right, I want you to pay attention to this now. This is very important. This is the implication. What is the relationship between the enzyme concentration, the substrate concentration, and the rate of reaction? So here's the very important graph. So this is very important. You please go and remember this. So what does it mean? What does this graph mean? Important point to note here is there's a fixed amount of enzyme. Okay, we're using a fixed amount. So given a fixed amount of enzyme, let's say we start off with 0 substrate. No substrate, of course, no rate. Okay, no reaction happens. Now let's slowly increase the amount of the substrate, the reactant. Okay, maybe let's put down reactant just to make it easier for you to understand. Right, so at this point, at low concentration of the substrate, the rate of reaction is directly proportionate. So it will be first order. Okay, why is this so? There's more than enough enzyme to accommodate the reactants. Okay, there's more than enough enzyme to accommodate the reactants. So the example I'll use here, imagine, okay, let's use this example. There are 5 car repair shops. Okay, 5 car repair shops. So these are the, you can think of these are the analogy for the enzymes. Okay, so they repair cars. Let's say you only have 2 cars. Let's draw, how do you draw a car? Okay, 1 car, 2 cars. Right? So there's a certain rate, the rate of repair. 2 cars, no problem. Right? So the number of the rate of car repair is proportional to the number of cars, right? So no problem. There's more than enough vacancies. Then you can have up to 5 cars. Okay, each car will take out a garage, or will take out a shop, repair shop. So up to 5 cars, no problem. The rate will be proportionate, right? The rate of repairs, how many cars are repaired per unit time, is proportional to the number of cars, which is these are the substrate. Now, however, what if we start to have even more cars? Then there's a problem, isn't it? There are more cars than available shops. So will it be proportionate anymore? Taken as a whole, it will not be proportionate anymore. Right? It will not be proportionate to the number of substrate. In fact, it will be less than proportionate. And that's exactly what we see here. Okay, this is essentially what we are talking about. So, at low concentrations, the sites are not fully filled, the enzymes are not fully occupied, and the rate is directly proportionate to the reactants. So if we first order, at higher concentrations of the reactants, the enzymes are all occupied already. Okay, very important now, fully filled. And any increase in the substrate concentration will not have any effect on the rate of reaction. I mean, the enzymes are all busy working already. I mean, there's no available enzyme to take up the slack, right? So the reaction becomes zero order with respect to the reactants. So you can see that here, this part is proportionate. Okay, directly proportionate. And this part is zero order. Okay, zero order with respect to the substrate. So you increase the substrate, also the rate doesn't change anymore. So you may wonder about this part, okay, because it will not be an abrupt change, it will be a gradual change. Okay, it's not a sharp line. And with this, you should be now ready to do question 10 of the self-check. Finally, the last part of this experimental methods for studying reaction rates, we will actually do some of these experiments in the lab. So you can take a look at color intensity, change in concentration, precipitate form, gas form. These are all things that we can observe. So you learn some of these methods where we do the kinetics experiment. Please go and read your lab manuals for more information on this. So a lot of it is we will do the experiments and you'll learn by doing the experiments. Okay, and with that, I would like you to just quickly go through this checklist to check your understanding of each of these concepts. Right, if you're not sure, then of course try the self-check, and of course attempt the discussion questions so that you're ready for the tutorials. Any questions you may have, you can ask on the Padlet. And with that, we're done with this series of lectures. Thank you for your attention.